Hey guys, so this is going to be a two-part series explaining how your body goes from a gene all the way to coding for a protein. This first part is going to talk just about transcription of DNA, and then there'll be a second video that will deal with the translation of DNA. So here we go. Hey guys, this is going to be a two-part series on what I like to call from gene to protein, and it's going to talk about everything that your body does to go from. So you know that genes are sections of DNA. They're sections of DNA that code for pretty much everything, for the cells and all of the parts and pieces that make up our bodies. They're all made from DNA. It's that same set of instructions. So we go from DNA, which holds the information, to proteins, which build the parts and execute the work, to cells, which build up to make an actual body. So this whole process is called, or this whole concept I should say, is called the central dogma of biology, and it talks about the flow of genetic information in the cell. So we know that DNA is replicating or self-replicating, so it's capable of making copies of itself. But when we take DNA and from it we make sections of RNA, this process is called transcription. When that RNA is taken and coded into a protein, that process is called translation. The protein itself codes for a particular trait that we showcase or we see, a phenotype if you want to think of it that way. So like I always tell you, DNA gets all the glory, but proteins do all the work. So it's almost through metabolism that we really and truly learned about genes. And one of the first things that they started looking at was the inheritance of metabolic diseases. Through this pattern, it suggested that genes code for enzymes. We knew that enzymes were types of proteins, so initially, genes code for types of proteins called enzymes. So the disease that we see is a phenotype. It is a trait, an expression of something, and it's caused in a disease by a non-functional gene product, also known as a non-functional protein or enzyme or something along those lines. So when we look at metabolic diseases, the disease itself is the phenotype. It is the result of a particular protein not being either produced or working properly. So these are just to show examples, and we've talked about Tay-Sachs, we've talked about albinism a bit. So those are just examples of metabolic conditions that are all caused because a particular gene, or a particular, enzyme or protein didn't work. So if you look at the pathway, you know that a pathway is made up of a series of steps, and we can't get to step B unless step A is complete. We can't get to step C unless step B is complete. Well, whenever we can't go from step A to step B, we end up with a disease. And the reason that these steps don't go from one to the other is because the enzyme that takes care of catalyzing this particular reaction is non-functional or not working. All right, so from gene to protein, we start off in the nucleus, which is where DNA is, and through transcription, we make a copy of mRNA as an example. There's some other types of RNA we can make, but we'll talk about those later. Then through translation, mRNA is read and interpreted, and we put amino acids together to form a protein, and then those proteins function or don't function to exhibit certain traits. So we start in the nucleus, we kind of end in the cytoplasm, and then throughout various parts of the body. So the big picture in transcription is that we're going from DNA language to RNA language. We're going from DNA to RNA. That's pretty much what we're doing, or what your body's doing, when transcription takes place. So let's refresh our memories about RNA. RNA does contain a ribose sugar. It does have nitrogen bases. It still has adenine and cytosine and guanine, but instead of thymine, it instead has uracil. It is single-stranded, and there are lots of different varieties. There is mRNA, tRNA, rRNA, and even a fairly recent discovery called siRNA. So in transcription, initially what we're doing is we're making RNA. Now in the kind that we will be looking at, we will be making mRNA. But be aware that rRNA and tRNA are all formed through the same process. So in transcription, for this particular purpose, we are making messenger RNA. And it's going to be made from one strand of DNA. So DNA unwinds and a side is chosen. The side that is transcribed or is copied into messenger RNA is called the template strand. The strand that is not copied or is untranscribed is called the coding strand. So the coding strand is going to have the same sequence as RNA because RNA is going to be complementary to DNA. We're using our base pairs. So A with U and C with G, just like we did before. They only pair up with one other kind of base. So that's going to make the coding strand, the untranscribed DNA strand, have pretty much the same sequence as RNA except for where there's U versus T. So the way for this to happen is through what we call a transcription bubble. So remember when DNA replicated, we had a replication fork. Well, now we have a transcription bubble. Pretty much the same concept. And there is an enzyme that is going to control all of the work or all of the action, and that enzyme is called RNA polymerase. So remember when we talked about DNA replication, the enzyme in charge was DNA polymerase 1, and it made DNA. Well, RNA polymerase makes RNA. So if we look at this picture here, outlined in pink, that pink bubble looks like bubble gum to me. That's supposed to represent your RNA polymerase enzyme. This is our double helix that we've unwound, so this is the section that we will be transcribing. This piece of DNA up at the top is not being coded. This is going to be our coding strand. So this is not the one we're transcribing. We're not copying this one. This one is called the coding strand. It is the strand on the bottom that we are coding, and this strand is the template strand. And then here you can see the piece of messenger RNA in green, kind of growing from that template strand. So there are 3 RNA polymerase enzymes. RNA polymerase 1 only transcribes rRNA genes, so it only helps with the making of ribosomes. RNA polymerase 2 is the one that we really care about in this example, because this is the polymerase that is going to transcribe genes into messenger RNA, whereas RNA polymerase 3 only transcribes tRNA genes, so transfer RNA genes. Now, they all work because of a section in the DNA, or they're looking for a section in the DNA that is called a promoter sequence. Those enzymes are going to recognize what we call the promoter sequence because it's going to tell them where to start working on the DNA. This is not replication. We don't need to transcribe the entire piece of DNA. We only need the section that codes for the protein that we ultimately want to make. So promoter sequences or promoter sections are just there to identify this is where that gene is going to start, and then there's something that will identify where that gene is going to end. So if you think about every series of codons, all of those letters, all of those bases making up a gene or making up several genes, which gene do we read? How does the machinery know which part of the DNA is going to be transcribed? Well, there's some features on the DNA that kind of alert the enzymes as to what's going on. I kind of think of it as the signs on, like your like the highway, for example. If I'm going to exit military, there's normally a sign a little bit before the actual exit that tells me that this particular exit is approaching. So I know, okay, this is where I'm going to get off. Well, it's kind of similar when it comes to genes and the enzymes that will work on these genes. The first important part is called the promoter region. The promoter region is a binding site and it starts before the actual gene. So it's at the very beginning of the gene. And what the enzyme actually does is it binds to a specific location, a binding site called the TATA box, TATA box. So we just call it the TATA box. And that TATA box is a binding site for RNA polymerase and its transcription factors. So they all bind there and they all get together and prepare themselves to go ahead and transcribe this section of DNA that they need. So it's kind of like the sign that says such and such exit in 0.2 miles. Or if you're listening to your GPS, they'll say so and so exit coming up in 1 mile or 8 miles or however many miles. And okay, now I need to kind of merge over into the lane that's going to take me to that exit. If I wait too long, I'm going to miss the exit. So it's kind of the same thing here. Without that promoter region, the enzymes wouldn't know where on the DNA to bind, and they would actually miss the gene that they're trying to transcribe. Another important region is called the enhancer region, and it is also a binding site, but it is far upstream. It's further up or away from the actual gene of interest. But a series of things are going to bind in that region, and it helps to turn transcription on at its most efficient level. So hence the name enhancer. It enhances the transcribing power of these enzymes and all of their parts and pieces. Okay, so what are these transcription factors? Well, you learned about them when we talked about cell communication. We talked about some kinds of proteins having to actually get inside of the cell and then binding to different receptor structures and being brought into the nucleus. Well, that's what these transcription factors are. They're an initiation complex. They bind to the promoter region, and there are a bunch of proteins that all bind to DNA, and they pretty much either turn on transcription or turn off transcription. They trigger the binding of RNA polymerase to DNA. So it's like, in the Caribbean, we have a saying. If you go to a restaurant and there's no one in line or no one's there, you don't want to eat there because obviously the food's not good. You go to the restaurant that has the longest line because that tells you that the food is really good there. So that's kind of how it is for these transcription factors in RNA polymerase. If there aren't a lot of transcription factors bound to the promoter region, RNA polymerase 2 has a difficult time finding the gene that it needs to transcribe. So it kind of looks for the crowd and decides that this is the restaurant I want to pick. It looks for the transcription factors and says, okay, this is where I need to bind. So then once it's bound, what happens? Well, it does very similar work to DNA polymerase. It just matches RNA bases to DNA bases. They're pretty much the same thing. The C's and the G's are going to go together. Wherever there is a T in DNA, there will be an A in RNA, kind of like always. But where there is an A in DNA, it's going to be replaced with a U in RNA because there is no T. So it's pretty much the same sequence. C and G go together, and A and U go together for RNA. All right, so once we've made a copy of our messenger RNA, it's actually what we call an initial copy. It's not really complete because eukaryotic genes are not continuous. In between the real genes, or the actual good sections that you want to make your protein, there are sections of junk, things that eventually your system and your machinery are going to have to get rid of. So the real genes are called the exons. Those are the parts that you want because they are actual coding DNA. And the parts that are not as important are called introns, the junk. So when you're done with that first copy, it's made up of a series of exons and introns, and exons and introns. So coding DNA and non-coding DNA, and it kind of runs the entire length of the copy that you've just made. So the saying that we have is that the introns come out, meaning you have to take them out, and the exons stay in, meaning you leave them alone. So what does it mean? It means that there's going to have to be some post-transcriptional processing or editing or tweaking if you want to think of it that way. We're going to need to take our piece of mRNA and fix it up a little bit before it really becomes messenger RNA. So the very first copy that we make is called the primary transcript, or the pre-mRNA. So again, this is going to be the first copy. And it is going to contain both exons and introns. If you look at this picture down here, this would represent that very first copy, the primary transcript, or pre-messenger RNA. And we're going to have to move some things around, so take some stuff out to actually turn it into messenger RNA. So what needs to be done is a process called mRNA splicing. And hopefully the word splicing makes you think of slicing or cutting things out, and that's exactly what happens. In splicing, we cut out the introns. We edit them out. We take them out. And once they've been removed, there's some other things that we're going to do to our piece of mRNA, but eventually we're going to make a mature mRNA transcript, one that we can actually send out of the nucleus to do its role. Now, your machinery is going to have to be really careful, because we're working with minuscule things you can't see with your naked eye, and now we're talking about taking the instructions of your body and pretty much chopping them into pieces. So splicing has to be a very, very accurate production. There's no room for mistakes, because if you cut off even one of those nitrogen bases, you're going to completely change what we call the reading frame. The reading frame is the series of codons, or the series of bases that make up a particular codon as the transcript is being read. So for example, if you look at this this top piece of DNA here, remember a codon is a group of 3 bases. So codon equals 3 nitrogen bases. So we'd have 1, 2, 3. That would be one codon. This would be another. Here would be another. Here would be another. Now see that section in red? The section in red is the nonsense information. It's the stuff you don't want. So that's the stuff we need to cut out. But if we make a mistake in cutting this, like for example, if we cut here, oops. See if I can get my pen working. Maybe not. Okay, if we cut there and we cut here, we're going to have a problem, because we will have 1, 2, 3. That would be a codon. And then these two plus this one will be a codon. So it would be a codon here and here. And then 1, 2, 3 would be another one, and another, and another, and another, and another. Well, look, we have a letter here that really should have been in a codon. It should have been in that last codon. But because we made a mistake when we cut out our nonsense information, we threw off the reading frame. And now this is left out, which means that this may not code for the amino acid that we actually need. And the picture goes on and shows you in detail exactly what I just went over. All right, so pretty much this top picture is, this is the piece of DNA, or the piece of mRNA, sorry. This is it properly spliced, and these are the amino acids it would code for. Here is the same sequence of mRNA. This is what happens when it's improperly spliced. So see, we have that extra G in there. That throws off the reading frame, and now we have a different sequence of amino acids. Actually, we get some amino acids that we really didn't want. So splicing has to be really, really precise and really accurate. So splicing is accomplished using a collection of RNA and enzymes. And they're really called RNA splicing enzymes. But collectively, we call them SNRPs. And it stands for small nuclear RNA and proteins. The enzymes are the proteins. So these guys will work together, and they will create what's called a spliceosome. So the spliceosome is made up of several SNRPs, and they have the ability to recognize what we call a splice site sequence. So it's a particular code that tells them cut here, and you'll have two of them, one on one side of the intron that needs to be cut out and one on the other side of the intron that needs to be cut out. So you'll have a bunch of SNRPs on one side and a bunch of SNRPs on the other side. They will make their incision and they will actually push the piece of DNA together. If you look at the picture here, you can see them doing that. And then they'll literally cut that intron out and cause it to anneal with itself, because remember, these ends are going to be complementary. And then they'll push the rest of the exon together and create a section that has just exons and no introns whatsoever. So it's almost like our cut and paste gene. They cut out the section that we don't need and they paste it someplace else where it can be utilized, or at least where it's not in the way. They're pretty cool, actually. So this actually is a really good way for your body to be highly efficient. And it does so through a process that's called alternative splicing. Alternative splicing is when you have alternative mRNAs that are produced from the same gene. So here's what you have to realize. That exons and introns aren't always gonna be the same exon and intron. So what I mean is, if you have an intron, like let's say for example, this blue piece right here is an intron. For the particular protein that we are trying to make, this may be an intron. But if we were trying to make a different kind of protein, this piece might actually become an exon. And it's because those pieces of DNA can play different roles that we can create multiple proteins from the very same gene, because the exons and the introns can be different depending on the protein ultimately that you are trying to make. So it's almost kind of getting difficult to define what a gene really is. We've always said that it is a section of DNA that codes for a protein, and that's still true, but what's doing the coding depends on what protein you're trying to make. Alright, so this is where we're going to break for transcription, and we will pick up with a different video for translation. See you guys soon!